Students, so today we will be talking about the topic of biological molecules. The past papers of biological molecules. We have already done the past paper practice of enzymes, of specialized cells, and of cells. These are three separate topics that we have already done the past paper practice of with the whole content coverage. First of all, I want to tell you one most important thing that the CI students, the Cambridge O-level students really make. O-level students are just like cranky or just focus on being lackluster and just focus and believe just they what they have memorized in the topic will just be asked in the MCQs or in the theory paper. They will say a student, and there is a student A which is a average student which has covered all the syllabus. He will won't be able to get A or A stars. It's impossible. He would may get a B or most probably a C, because there is a difference between an average student and a smart student. Average student thinks that what they have learned in their lessons, what they have learned in their syllabus coverage, and they have all done the rote memorization and everything, but they don't understand that Cambridge don't believe in rote memorization. The MCQs only focus on concepts. If your concepts are clear, if you have a great understanding skills, if you have a great command over English, and that will only come when you do press paper practice. So I want you guys to attend all the past paper sessions, because past paper sections makes you more competent so you can answer the questions, because examiner won't be asking you and won't be giving you marks on the question like what is your name, my name is Shayan, you will say, and you will get a A. That's not the case. Examiner wants you to know the content and apply it on certain questions. Like if there is a question on food test, you should know full information about enzymes before doing biological molecules. That's why I did enzymes before biological molecules. Why did I did it before the biological molecules? Because you should know what type of biological molecules will be digested by that certain enzyme. So you should have a whole command over the content, and you should have a good reading skills. So there are two important things that we all gotta understand, that is our reading skills, that we are reading four words or a full stop till sentence, and then we are analyzing or evaluating what the question is saying to us. Or the second thing is your understanding skills, what the question or what the examiner wants you to answer. Like if the examiner is asking you a food test, you won't be writing about the carbohydrates. If the examiner is asking you about reducing sugar, you know the difference between reducing sugar and the starch solutions. You should be knowing the difference between each of them. So let's jump on the topics. Let's jump on the topic. The first MCQ is a bit tricky for those who haven't been doing past papers. It will be tricky. I will explain it in the most easiest way possible for myself. All right, this is the question from May June 2022. All past papers questions. The experiment shown was set up and left for 30 minutes. So you should be knowing that experiment was left out at least for 30 minutes. This experiment was let out for 30 minutes. The the membrane is permeable to sugar and water, but not permeable to starch or protein. What is the meaning of permeable? This is where you apply your content information. This is the point where you apply your understanding skills. So permeable means, like, it is saying permeable means partially permeable. Things can go out and things can come in, but certain things can come in and certain things can come out. It's not fully permeable. Fully permeable means everything can go out, everything can come in. That is fully permeable. Partially permeable means certain things, like only protein can come out, but starch cannot. Only water can go in, no salt can go in. That is partially permeable. And what is completely permeable or fully permeable? Everything can come in and everything is can go out. So not permeable to starch or protein. So this means starch and protein cannot come in or cannot get out. After 30 minutes, the samples of the water in the beaker were tested with a Bannatyne reagent, burette reagent, and iodine solution. Now, here is the point: you apply your information that you have. Bannatyne salt reagent is for starch. Bannatyne solution is for all right. Bannatyne is for what is known as reducing sugar. Reducing sugar. Burette reagent is for protein, and iodine reagent is for starch. So there are three processes going out at the same time. All right, the experiment shown the setup. First, let's read the labeling. It is partially permeable, starch cannot go out, or starch protein cannot go out. There is water. It is not a salty water, it is just pure form of water. Mixture of starch and amylase solution. So the beaker has starch. Starch is what? Polysaccharide molecules. It is a reduced, non-reducing sugar. Starch is a polysaccharide. When this polysaccharide is put with amylase, now comes the information for the enzyme chapter. Amylase is an enzyme that destroys the polysaccharide molecule, which is starch. Starch is digested by amylase. Starch is what? A non-reducing sugar. So starch is broken down to maltose. Starch is broken down to maltose, and that maltose is a disaccharide molecule, which is a reducing sugar. Let me repeat. It is saying that in this pouch, which is partially permeable, so water can come in, mixture of starch and amylase solution were left for 30 minutes. In this duration, the enzyme breaks down the starch molecules in which is a polysaccharide, which is also a non-reducing sugar, which means it does not react with Bannatyne reagent. It converts into a reducing sugar, which is maltose, which is a disaccharide. So let's now see. So now it is a reducing sugar. So reducing sugar will show the positive results. So there is reducing sugar. So it will convert from blue to green or yellow or orange or brick red. It can convert into these four colors. Let's see. Bannatyne test is for reducing sugar. There will be no there will be a test. It will convert into from blue to red. So these two can be the answer. Burette test is for what is known as protein, but there is no protein. A churner permeable sugar, starch, and protein mixture of starch and amylase. There is no protein over here. So I would say there will be no color change. What is the color of burette? Blue. I told you a mnemonic to memorize. What was that mnemonic? B for burette, B for blue, P for protein, P for purple. So if there was protein, so the color must have changed to violet or must have changed to purple. But it does not change and it stayed blue. Why? Because burette was the blue color and there is no protein in the solution. So I would say these two can be the answer. Now iodine test. Iodine test is for a non-reducing sugar, which means it is for starch. But this is for starch. Iodine test is simply for the test of starch. But here, the examiner tricks us by saying amylase. If a student who has done rote memorization has no understanding skills, just put a tick on the C. Because he don't understand anything, he is totally blank and out of space. He will text on the C or the B just roughly. He don't know. But you should know the question is saying amylase solution. When it's saying amylase solution, which means amylase digests starch into maltose, and maltose is a reducing sugar. So starch is not present, so there will be no color change. Iodine solution is what color? I told you two mnemonics, one for protein, one for starch. Starch mnemonic was yummy brown blueberries, yummy brown blueberries, yummy brown blueberries. Yummy means yellow, brown to blueberries, blue, black. So yellow, brown to blue, black. Yellow, brown to blue, black. So there will be no color change. Why there will be no color change? Because there is no change. There will be no color change. Why there will be no color change? Because why? Because there is no starch present in the test in this beaker. So B is the correct answer. Let's go on the second question. See the amount of times examiner has asked this question more than 5 times. That's a really impressive thing because examiner don't really repeat questions whether in any of the subjects, but it does. Four food tests were each are carried out on the four different white powders A, B, C, D. The results are shown in the table. Which powder contains a carbohydrate, a protein, and a fat. All right, four test tubes were each carried out on four different white powders. There were four different white powders, and there were food tests on them. The results are shown in the table. Which powder contains a carbohydrate, a fat, and a protein? All three both. All three are present. So results of the test of the powders. Benedict test was blue color. Blue means there was burette, B for blue, B for burette. So there was no color change. So A cannot be the answer. A can be the answer, we can say, because let's read the other questions. Orange color, Benedict's test, orange color. Then we have red color, and then we have green color. Benedict test is for reducing sugar, and a reducing sugar is what? A carbohydrate. Reducing sugar and starch are two different types of carbohydrates. There are two simple types of carbohydrates. So if I'm saying reducing sugar, or if I'm saying what is called as uh, if I'm saying reducing sugar or I'm saying starch, it means the same because starch and reducing sugar are the two different types of carbohydrates. So reducing sugar color change goes from blue to green to yellow to orange and to brick red. So if I'm saying Bannetic test was carried out, so it should be changed from blue to green to yellow to orange to brick red. All right. It changed from orange color, changes to orange color, it changes to red color, and it changes to green color. Alright, burette test is for protein, P for protein, P for purple, B for blue, B for burette. So burette is of violet color, there is a color change. A can be the answer. Blue, no, blue is not a color change. There is no color change. So C, B, and D are cancelled. It could either be A or it could either be C. Why there are only two possibilities? Because if there was a protein present, it should change to purple, which is also a shade of violet. So it should have changed to violet or purple if it has a protein. An examiner wants us to tell which one has protein in it. So violet color, these A and C could be the answer. Ethanol emulsification test, it should have a milky solution forming over the solution. So A could be the answer. Or B, but B was cancelled, so we cannot say B is the answer. We could not say D is the answer. So it is A. I will go even further. Iodine test was carried out, which was blue black color was formed. A is the answer. We all know blue black color was formed. Let's say iodine test was carried out. Iodine test has four possibilities: yellow, brown, changes from blue black. So it changes to blue black. That's the correct statement, and A is the answer. I will repeat myself over and over again. Why is A the answer? A is the answer because the examiner wants us to tell which of the powder has both protein, has both fat, has had have starch. There was a iodine test carried out which changes from blue black color, which means iodine test was carried out, blue black color was formed, yellow brown to blue black, yellow brown to blue black, which was a iodine test. Examiner wants us to know which one has carbohydrate. Iodine starch is a type of carbohydrate, and starch food test is iodine solution. So when iodine test is carried out, there should be a color change if starch is present, and examiner wants us to tell which one has starch. So blue black color was formed, so which means starch is present, because most of the time it changes from yellow brown to blue black, yellow brown to blue black. So there is a color change. So this means the iodine test has shown a true result. There is. So blue black done. Ethanol emulsification test. If fat was present, there should be a cloudy like substance formed. So milky thing is formed. So this means fat is present. Why fat is present? Because ethanol test is only for fats, and fats, when for fats is found in any solution, any powder, anything, there should be a milky substance found. Burette test is for protein, and if there is a protein, the color change will be from blue to purple. If there is a Benedict test, it should change to blue, green, yellow, orange, and it does change to blue. So which means A is the correct answer. All right, identifies the chemical compound in proteins. We know protein is made of carbon, is made of hydrogen, is made of oxygen, and is made of nitrogen. There are some MCQs which just want you to know the content, but most of the times there are only the MCQs which just want you to have understanding skills. So we would just look for the options where there are all ticks. A has all ticks because protein has carbon, protein has hydrogen, protein has oxygen, and protein has nitrogen. So A is the answer. Let me highlight it. So A is the answer. Let's move down to another question, which is only asked once. Which law shows the chemical elements contained in fats? Fats has, let me first change the pen color. Which law shows the chemical elements contained in fats? Fats is has the same chemical combination as carbohydrates, where at it has a more ratio of hydrogen. If you are not able to understand any of the thing I'm saying, go to my video and watch about fats and every biological molecule video, or go to the playlist. I have made a playlist only for the biological molecules. Go watch it before watching this video, because you won't even understand a hack of this biological molecules. So there is fat present. If there is fat present, it has the same chemical combination as carbohydrates. So there should be carbon, there should be hydrogen, there should be oxygen, because in the ratio of 2 C's to H12 and O2, this is the combination for which is so called as glycerol. So there is no nitrogen, so there it A should be the answer, because carbon is present, hydrogen is present, and oxygen is present. Simple question, no understanding thing needed. All right, a student carries out a test to see whether the solution in the test tube contains protein. If there is protein, there will be a color change from blue to purple. If there is a color change, then it has protein. Which row shows the correct food test and a positive result? It is saying the positive result, which means we have to see where is blue to purple. So burette is the test for protein that we all know. If you don't know, go watch the video of instant test biological molecules food test. So burette, it should be, it is blue black and purple. Blue black is not the color change, purple. So purple, D is the answer. Why we are it, the examiner wants us to know where is the positive color change, where is the color change. So the color changes from blue to purple, that we all should know. It has a burette test, C or D could be the answer. But when it's saying positive test, which means it should be from blue to purple. Blue black means there is no color change, there is no protein, but it is saying there is protein, and there you should know the positive test. So D is the answer. Now let's oh my god, see the amount of times examiner has asked this question, more than 6 times. Oh my god. Small molecules are used as the basic unit in the synthesis of large molecules, blah blah blah. We just need to choose the statement which is correct. See, there is a smart student and there is a brilliant student. I am keep on giving these examples. There is again a brilliant student and an excellent student. The only difference between the excellent and the bright student is how he solves the MCQs. A brilliant student will see a difficult question and he will skip it. He don't want to ruin his paper. If he has saw a difficult question, oh my god, I have never studied this, my paper will be bad, my result will be bad, my parents will be depressed, everything he bad listens, and his paper goes very bad. Just really just say very bad. His paper goes really, really bad because a smart student always thinks about his results, about his families, about his everything. He ruined his career, he ruined his paper himself. That one MCQ doesn't means that your whole paper has ruined. And even that MCQ is also easy, just your brain is playing with you. Just the examiner is playing with you. A brilliant student will leave that MCQ and at the end of the paper, he will try to solve it. And if he doesn't understand anything, he will just hop on the last statement of the question after the full stop before the question mark. He will read that statement and determine either that statement what it wants us to know. I first of all, let's see, I didn't even understand this question. I just skipped it. I don't know this question. I skipped it. I don't even know a hack of this question. I will just hop on the statement after a full stop and before a question mark. Which statement is correct? We just want to know which statement is correct. We don't want to hack anything to do with this. So a brilliant student will always keep focused and will always think what is the right answer. He will just hop on the MCQs A, B, C, D, and he will eliminate the wrongdoing which doesn't make any sense. Let's see this question. Let's see what a brilliant student will do. Amino acids are the basic units of carbohydrates. Does this make any sense? Does this make any sense? Like carbohydrates are made of amino acids. That's the most strongest thing we have ever heard in this 3 years journey. Amino acids makes protein, not carbohydrates. So this is the most simplest statement that we could state as incorrect. The second statement, fatty acids are the basic units of glycogen. That's the most strongest thing I've ever heard. In the beginning of the video, I told you there are two types of carbohydrates. The first type is the reducing sugar, and the second type is the starch. And the starch is another. There is another type of starch which is glycogen. Glycogen is also like starch. So glycogen is carbohydrates, not fatty acids. So this is the eliminating statement. Glycerol is a basic unit of oils. That is correct because glycerol is fat. If you don't know, just skip this MCQ, read the second one. There, we just know that glycerol is fat. There are two types of fats, glycerol and fatty acids. If you haven't watched my video, go and watch it. There are two types of fats, glycerol and fatty acids. Fatty acids and glycerol are oils. There are two types of oils, saturated ones and unsaturated ones, that I discussed in my video. None of the MCQ is out of my videos. Simple sugar is a basic unit of protein. No, amino acid is the basic unit of protein. If you don't know glycerol, if you don't know oils, if you just eliminate these three options correctly, you will be left with the correct one. That is what a brilliant student does. He just goes for the right and for the common sense one. Four solutions were tested to see which whether they contain protein, starch, or glucose. The colors of the solutions after the food test are recorded in the table. Which solution contain protein and glucose but does not starch? They want to know which one contains protein and glucose, not starch. Glucose is another type of it is another type of starch, but I don't know what they want to. Glucose is another type of which is known as a reducing sugar. Let's just hop on. Buret test, there is a Buret test, 7 question. There is a Buret test. There should be a positive result for protein. And what is the positive result? Blue to purple. So these two are the wrong answers. It would be either C or it would be either D. Iodine test, it is for the iodine test is for starch. There should be no result, no color change, because the examiner is saying it don't want starch, and starch test is basically what? Brown, which is basically iodine test. Iodine test is for iodine test is for starch, but the examiner is saying he does not want starch in his solution, so the color will stay same. So brown, yellow brown to blue black. So it this won't be the answer. C is the answer. Banetic test, there should be a color change because examiner wants glucose, and glucose is a reducing sugar, and reducing sugar test is Banetic test. Banetic test is a reducing sugar test, and the reducing sugar is glucose is a reducing sugar. So this means there should be a color change orange, because blue, green, yellow, orange, and brick red. So there should be orange color. So C is the correct answer. Let's go on this topic. If you don't, which reagent is used when test food for vitamin C, vitamin D's test is. If you don't know any, haven't studied about vitamin C, whereas I have taught vitamin C, but just suppose you don't know. So just see the other options. Iodine test is for reducing sugar, so this cannot be the answer. Is for starch, so this cannot be the answer. Ethanol is for fat, so this cannot be the answer. Banetic solution is for reducing sugar, so this cannot be the answer. DCPIP, whereas is there is no other option left, so DCPIP is the answer. You have to know the how to eliminate the wrong answers. Which statement about biological molecule is correct? If you don't know the anything, just go for the elimination. Proteins are made from fatty acids. No, proteins are made from amino acids. Most simple things. Oils are made from amino acid and glucose. No, oils are made of C2H12O2. So a protein, whereas is made of amino acids. Glycogen is made from glycerol molecules. That's wrong. Glycogen is made of disaccharides molecules. Is made from monosaccharide molecules. When small monosaccharides molecules join together, they form glycogen. So that's the wrong. If you don't know glycogen, please watch my video. You will understand. And DNA contains the bases A, C, G, T. That is right. We studied that DNA is the most difficult topic in biology for students, but I did it in only 13 minutes. You gotta watch it. DNA has 4 bases, and there are 4 bases. We wrote the mnemonic. There is CAGT. So this is the right answer. A is the right answer. Let me mark it. Let me go, dude. All right. Acha, the data shows the concentration of sugars and starch in an onion. There is reducing, there is a sugar and there is a starch. So there are two reducing sugar and starch. There are 3.5 and zero starch. There is no starch. There is only reducing sugar, which means there is no starch. The onion is tested with a Benedict solution and an iodine solution. Iodine solution is again for starch. Reducing sugar is again for reduce. Benedict test is for reducing sugar. Which set of result is correct? There should be no color change in iodine solution because iodine solution is for starch and there is basically no starch. So there should be no color change. So it will stay brown. It will stay brown. So these two can be the answer. Whereas Benedict solution should show a positive result because it has a reducing sugar. It has 3.5% of reducing sugar, 3.57 grams of reducing sugar basically. So reducing sugar test, we memorized a mnemonic. We memorized a mnemonic: blue to green, blue green to green, yellow, orange, brick red. Green, yellow, orange, brick red. Green, yellow, orange, brick red. So it should be brick red. C is the answer. Oh, sorry, D. Because this should match this answer. So, sorry, D is the answer. All right. On the basis sequence of part of one strand of DNA molecule is shown, which is the base sequence of the other strand. I told you a mnemonic where teachers don't tell you any mnemonic. That's why you fail to answer these questions because there is a pile of questions more in the syllabus. You forget the mnemonics and everything. So I created a simple mnemonics. Simple mnemonics. The simple mnemonics were teacher. Let me write. Teacher assistant. Teacher assistant. Just leave the spellings. I will correct it later on. Teacher assistant and Google. You all know Google. Uh, don't joke with me. Google Classroom. This is the mnemonic. Why I'm saying this is the mnemonic? Teacher T, assistant A, teacher T matches with A and A matches with T. G is Google, C is classroom, G matches to C and C matches to G. There is a simple mnemonic: teacher assistant, TA or AT. There is a simple mnemonic: Google Classroom, GC with G will matches to C or C will matches to G. Teacher assistant, T will matches to A or A will matches to GT. So A, T, let me zoom in. I hate this thing. All right. All right. It is. Wait a minute. All right. All right, we have studied A will match teacher assistant A to T, teacher assistant T to A, teacher assistant A to T, Google Classroom G to C, Google Classroom C to G, Google Classroom C to G. So this would be the answer. So this where is the answer? All right, T T A T C G G. So this is the answer. Simple. There is nothing for rote memorization. You just gotta memorize the mnemonics and the understanding skills. That's it. The apparatus shown in use for an experiment on starch digestion. Which test tube contains the most sugar after 20 minutes? You should first know the labelings, all the labelings, and the 20 minutes. All right. First, let's focus. There are two solutions: starch solution and with salivary amylase. Amylase will digest starch. Amylase is an type of enzyme, and amylase enzyme digest starch. So starch will be digested, amylase will digest it, and that's simple. Starch will be digested by amylase. So there should be no color change. A cannot be the answer because starch is digested and there is nothing left except water in it. All right. There are two types of starch solutions in B and C. They are both same. There should be some difference. I will see. All right. There is now D, starch solution and salivary amylase. All right. All right. Salivary amylase will digest D. So D, there will be nothing left in the D. Now we are saying B and C, the practice shown and the starch on starch digestion, which test tube contains the most starch in 20 minutes. It's asking which test tube won't have starch, no starch after 20 minutes. So these two cannot be the answer because there isn't any amylase in them. There are no amylase in them. So this cannot be the answer. A can be the answer or D can be the answer because they both have amylase and the amylase has digested their starch. So they have no starch. Let's now figure out either A will have more starch or B will have more starch. Here we see temperature 15 Celsius and 37 Celsius. We studied the enzymes work at the most optimum. Enzymes work more on their optimum level. So when they have optimum temperature, they will work more. So 37 is the best temperature for enzyme activity. So enzyme will digest starch quickly. So D is the answer. Simple. All right, which statement about the DNA structure is correct? All right, base A pairs with C. All right, let's write AT, teacher assistant, GC, Google Classroom. A matches to T. Yes, that's right. A matches to T. Or T matches to A. T matches to A or A matches to T. Google Classroom, G matches to C or C matches to G. It consists of proteins. No, enzyme DNA does not contain some proteins. It forms a simple whatsoever. We just know B is the answer. All right. Let's all right. Which process depends on the fact that water is a solvent? Which says that water is a solvent, which process depends on the fact that water is a solvent, water is solvent, alright, blah, 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 blah. Evaporation from a spongy mesophyll cells and blood plasma, this contains from the, which is known as movement in and out of cells. So we will be skipping this MCQ. We won't be doing this MCQ right now. We will be doing it in the past paper session of movement in and out of cell. The table shows the results of food test carried on out on a fruit. It is a test which has carried out on a fruit which says Benetic is for reducing sugar. So there is a positive result which means reducing sugar is present. Burette test is for Burette test is for Burette test is for protein. So this means protein is present. Protein is present. Ethanol test is negative. No fat is present. Iodine test is for starch, which means starch isn't present. So these two are failed tests, which means there is no fat. There is no fat and there is no thing called as starch. There is only reducing sugar and there is only protein. So, which that the fruit contains fat and reducing sugar? No fat. There is no fat. There it contains fat and starch. There is no fat again. So B cannot be the answer. Protein and reducing sugar. That's right. It has protein and it has reducing sugar. C is the answer. Protein and starch cannot be the answer because it has no starch. All right, four test tubes were set up as shown in the table. In which test tube would starch be broken down the faster? 2 cm cube starch suspension added, 1 cm cube of amylase added, 1 cm cube of boiled amylase added. Boiled amylase means the amylase won't work. Amylase won't work. Why amylase won't work? Because it is boiled enzyme denatured. Temperature C, in which test tube starch will be broken down the fastest? If the enzyme at the point the examiner say on the boiled amylase, no result will be there. Why? Because when the enzyme is boiled, there will be nothing called as enzyme. There will be a irregular structure. Enzyme shape will be changed. So if they want starch to disappear, there should be ticks over here. So C, D could be the answer because this cannot be the answer. 1 cm cube of amylase added. Why? All right, amylase is added. Cm cube, 16 cm cube of which is called as. All right, amylase fastest. All right, B is this can be the answer. B can be the answer because there is no there sorry there this cannot be the answer because there it is added. There is no mean of adding a boiled amylase which is already a dead amylase. So it could be either C or it could be either A or it could be either B. All right, so 1 cm cube of amylase added, it could be because amylase digests, so this could be the answer. 2 cm cube of starch added, so starch can be digested, this could be the answer. Only depends on the temperature. In my previous MCQ, I told you enzymes work on their best when they have optimum temperature. So there is the optimum temperature. 35 is the optimum temperature. Here it works. All right. 16, when a food substance is added with Benedict test, food substance is there, which shows the presence of starch. So there should be iodine is of yellow brown, so there should be blue black. So it should convert into blue black. A is the answer. All right. All right. The diagram shows the structure part of a DNA molecule. What does X represents? This is, I have shown you, this is a hexagonal structure. Here is nitrogenous base and here is phosphate group. And when this attaches with another nitrogenous base of another hexagonal structure, this is how a this is how a DNA looks like from inside, and here it is how it is represented in this diagram. So this is a base because this is base. This is what represented over here. So here's the base carbohydrates. No, there's no carbohydrates, there's no protein, no amino acid. If you don't even know what is base, just eliminate the wrong answers and you will get the right answer. What is the color change shown on the Benedict test when heated with a reducing sugar? What is the color change of by Benedict test and with when heated with a reducing sugar? There's a reducing sugar which was tested with Benedict test, which is the means there should be a reducing sugar. All right, there should be a color change. There should be a positive color change from blue to green, yellow, orange. So blue to red. All right, blue to green, yellow, orange, brick red. So orange, brick red is the same thing, red or brick red. Which diagram shows the correct stand of DNA? All right, this is wrong because I told you A, teacher assistant, TA, teacher assistant, or GC, Google Classroom. So I told you the mnemonic, just follow this mnemonic. AT, no, this cannot be the answer. AT, this can be the answer. AC, this cannot be the answer. AG, this cannot be the answer. All right, B is the answer. Done. All right, the bases on one of the strands of DNA molecule have the sequence shown A, C, T, C, T, G. What is the corresponding sequence of the bases on the other strand? A attaches to T, this can A attaches to T, T attaches to A, C attaches to G, T attaches to A, and G attaches to C. So D is the answer. Simple. Acha, which element is found in proteins but not carbohydrates? Carbohydrates has carbon, hydrogen, oxygen, whereas protein has carbon, hydrogen, oxygen, and nitrogen. No nitrogen is found in carbohydrates. So C, nitrogen is the answer. Simple understanding and content memorization. When bases pair in the formation of DNA, what is the one of the pairing? G attaches to C or C attaches to G. T attaches to A and A attaches to G. How to memorize this? Google Classroom and Teacher Assistant. So G attaches to C, B is the answer. G with C, Google Classroom, or G with T, GT, no. Teacher Assistant is not there. Teacher Assistant, no. So there is, I think, the pair basis of the formation of DNA. So B is the answer. All right, that the diagram shows the order of along the part of the strand of DNA. There is three and a half there in the four. Out of 4, there are 3A, there are 3A, 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 all right. The correct order of the bases of the corresponding which they attach to, corresponding means which they attach to of the other strand A to T, C to A. Google Classroom, no, this cannot be the answer. T to A to T, C to G, T to A, teacher assistant, T to A. So D is the answer. All right, let's do it still here. And this is the, I think, the last. The diagram shows a section of DNA with 4 bases identified on the stand. So G attaches to C, A attaches to T, C attaches to A, Google Class, Google, sorry, G attaches to C, C attaches to G, and T attaches to A. That's it. We will do the other MCQs with the theory part. Thank you for watching this video, guys. Just remember, you have to focus on understanding and reading skills, not just on the content memorization. Do past paper practice. Do watch my videos. Bye, guys.